the next type of the cofactor is the prosthetic group. Prosthetic group is an organic component, and remember, prosthetic group is different from the coenzyme. How is it different? Both are organic components, but they are different. Prosthetic group is tightly attached to the apoenzyme. So they are tightly bound. That means they don't separate every time a reaction going on. They will remain attached permanently, and that means they are tightly attached. And coenzymes are those factors which loosely attaches to the apoenzyme. That means they attach only for reaction and then they separate. They attach again with some other apoenzyme and then separate again. So they have a permanent bond and they change from apoenzyme to apoenzyme. They keep changing because they are loosely attached to them. Example, heme is the prosthetic group of catalase enzyme, peroxidase enzyme. We have learned these enzymes already, if you remember, in the peroxisome topic of the cell organelles, or you can say 8th chapter. We have already covered these enzymes function there. So please check it out from there, the function of these enzymes. Then the loosely attached type of the cofactors are called as the coenzymes. And coenzymes are many. The easiest examples are NAD+, NADP+, and coenzyme A. We'll be learning about them again in the respiratory chapter. And these are required for dehydrogenase enzyme. Which enzyme? Dehydrogenase enzyme. So let's try to understand some basic difference between prosthetic group and the coenzyme. You can say prosthetic group is a non-protein component of conjugate enzyme that is firmly attached or tightly bound to the apoenzyme, and the coenzyme is the non-protein component of a conjugate enzyme that is loosely attached to the apoenzyme. And then because they are tightly bound, so one more point is possible, that the same apoenzyme is used in picking up and handing over small chemicals. Because they are permanently bound, so same apoenzyme will be used for every reaction by the apoenzyme. Because they are going to have a permanent bond, so they don't change. In this case, because they are loosely attached, today if in a particular reaction the coenzyme is attached to the apoenzyme, in the next reaction it will attach with some other apoenzyme. Reason is but loosely attached. So different apoenzymes are used in picking up and handing over the small chemicals. So they have a one-to-one relation and they keep changing their apoenzyme from reaction to reaction. Now in the same manner we can understand differences between apoenzyme and cofactors also. Apoenzyme is the protein part, coenzyme is the non-protein part. Apoenzyme is larger in size, the coenzyme is smaller in size. Apoenzyme is thermolabile, coenzyme is thermostable. Apoenzyme does not participate in the transfer of the chemical, but coenzyme has the property of picking up and then transfer the substance from one substrate to the other substrate. Apoenzyme is specific for an enzyme, but cofactors are variable. We already learned with this point. Apoenzyme is fixed for every enzyme, and then the coenzyme may function as cofactor for many other enzymes. Then remember, apoenzyme is involved in catalytic activities of enzyme, but coenzyme's role is what? Picking up the product of the reaction and handing over the same to the other substance. So catalysis is done by the apoenzyme and transfer is done with the help of the cofactors. Now some enzymes are there which are synthesized in the inactive form. If you remember in your 10th class, digestive tract topic, stomach topic was there, and you were always told that there are enzymes which are formed in the non-working manner, and then the reason is they can cause harm to the human body, and when conditions are conducive or correct, they will become active. So such enzymes which are synthesized in their inactive form are called as zymogen. Now pay attention, same thing, zymo. Gen means to produce. So something which will give rise to the real enzyme, that means something which is the precursor of enzyme, is called as proenzyme. So proenzymes are inactive. Proenzymes are enzyme precursor. Precursor means something which give rise, a raw material, which will with one step reaction will give rise to the real enzyme or active enzyme. Remember, these enzymes need a specific pH, they need a specific substrate, or then only they become activated. Now remember their size now, because obviously they are inactive, so that means they will be bigger in size compared to the real active enzyme. So what exactly happen is, the inactivity causing portion is removed by maybe HCl, maybe some other enzyme, and then finally they become active. So I have written 3 examples. First is the pepsinogen. We all know pepsin is synthesized as the gastric juice enzyme. So pepsinogen is activated by the presence of HCl. HCl will turn it into pepsin, and pepsin is protein digesting enzyme in the stomach. In the same manner, trypsinogen comes when in the topic of your pancreatic enzymes. So trypsinogen is activated by trypsin. So in your intestine, when digestion is going on, trypsinogen has to be converted into trypsin. This happens because of the enterokinase enzyme, which is released from intestinal juice. Enterokinase convert trypsinogen into trypsin, and remember it also has autocatalytic activity. Once a small amount of trypsin is formed, it will convert all other leftover trypsinogen into trypsin. So even trypsin can cause formation of trypsinogen, conversion of trypsinogen into trypsin. In the same manner, chymotrypsinogen, same thing, is converted into chymotrypsin with the help of the trypsin enzyme. So this is how some enzymes become activated. From here onwards, we will continue in the next class.